Hello, now welcome to the module 3.1b of the MOOC on Bioinformatics Essential. In this module, we'll learn about foundations of sequence alignment, more about this thing. So, introducing gaps. What are the gaps in the alignment? Biological sequences can evolve through insertions or deletions. Okay, so whatever evolution we have seen, or whatever evolution we have studied so far, it is due to the insertions and deletions in the sequences. To represent these events, alignment algorithms may introduce gaps. Okay, so see here, sequence A, it contains ATGC dash ATGC, and sequence B contains ATGC C ATGC. Okay, now this dash represents a gap, and this gap allows remaining residues to be aligned in a biological meaningful way. Now, why would a gap be needed? Now consider these two sequences ATGCTA and sequence B ATGCXXTAGCTA. Now this second sequence contains an additional region. So without introducing a gap, corresponding positions may become misaligned. So a gap allows the alignment to compensate for possible insertion deletion events. Biological, we can say that a gap does not necessarily mean that the sequence is wrong. Okay, it may represent an evolutionary insertion or deletions. In short, the gap in the sequence, it shows the insertion or deletion, and it shows the evolutionary linkage between the that two sequences. Mismatch and match, mismatch, and gap. So an alignment generally evaluates three basic situations. So gaps has the three basic situations. First is match. Match is AA, means same residues. Okay, if there are same residues, that is called as the match. For example, you can see here first sequence is A and second sequence is A. Means the first letter in sequence A is A, and first letter in the sequence B is also A. That is called as the match. Same residues, that is called as a match. Mismatch means different residues. If sequence A contains A and in the same position sequence B contains G, that is called as a mismatch, different residues. Gap is if sequence A contains A but sequence B doesn't contains anything, that is called as the gap. Okay, and that shows whether sequence A has some insertion, or we can say that sequence B has the some deletion relative to other sequences. So alignment algorithms use the scoring systems to determine the most appropriate arrangements. Now, alignment score basic concept. Suppose alignments. Now we just seen that alignment has the three gaps. Okay, three situations: match, mismatch, and gap. Okay, now match, if there is a match, then we will assign some positive score. If there is a mismatch, we'll assign some penalty, and if there is a gap, we'll assign the penalty. Now alignment algorithm searches for an arrangement that produces a good score according to its scoring system. How, I will tell you. Simple conceptual model. This is not the actual model. Okay, we are just doing the illustrations. These values are just the illustrations. Okay, so suppose match, if there is a match between two sequences, one for one position will assign +1 mark. If mismatch between two se- uh, between two nucleotides of the sequences, okay, at the same place. If there is a mismatch, we'll assign -1 mark, and if there is a gap, we'll assign -2 marks. Means if there is a insertion or deletion, we'll assign the -2 marks. Now these values are only illustrative. Just make sure that these values are the illustrative. Actual bioinformatics tools use more sophisticated scoring systems. They have their own scoring systems. Okay, we will not go in much deep of that. Just make sure that if there is a match, +1, if there is a mismatch, -1, and if there is a gap, -2. These illustrations. So based on the how many matches, based on the how many mismatches, based on the how many gaps, we do this maths in the background, and we show that, and we find out some score. Now, coming to the main thing, why this scoring matters. So consider two possible alignments. Alignment A has the many matches, few mismatches, few gaps. Okay. And alignment B has the few matches, many mismatches, and many gaps. So based on these, we can calculate the score. Okay. And the scoring system helps to determine which alignment provides stronger evidence of correspondence. Okay. So where we are getting the stronger evidence, and this is why the alignment is not simply put the sequence underneath each other. Okay, this is not the thing like only two sequences under each other and we are doing something maths. And okay, it is the computational optimization of problem. This thing is very important. We have to optimize the problem in a computational way. So that's why this scoring is matter over here. Now, global and local alignment in the blast context. Now the connections to blast becomes much easier. Blast is primarily designed around local sequence similarity searching. Instead of asking, can the entire query sequence be aligned from beginning to end with every database sequence? Blast asks in a simplified way, are there statistically significant regions of similarity between this query and sequences in databases? So instead of making entire sequence to align it and find the homology analogy between them, this is not the way here. Simply in blast, what we do, we find the significant regions of similarity between our query sequence and the sequences in databases. That's it. So this is particularly powerful for database searching. Why blast uses local similarity? Now see, consider one protein. Protein has one unknown region, one conserved enzyme domain. Conserved enzyme domain means this domain is common in every or many in the many species, we can say. And then unknown region. So the complete protein may not closely resemble any known protein, but the enzyme domain may be highly similar to known protein. So a local similarity search can detect that conserved region. This is one reason BLAST is so useful for biological sequence analysis. So here we just find the similarity between this conserved unit to another species, so that we will get what is the conserved region, and then based on that we can predict the function of another regions and so on. So this is the reason. So now one small difference here, pairwise alignments. Here we compare two sequences, example human protein versus mouse protein. Simply two sequences we do pairwise alignment, pairing. And sampling technique, not a sampling technique, here it should be the multiple sequence alignment. Okay, so here we do the three or more sequences. Okay, for example, if we compare the human, mouse, rat, dog, chimpanzee here, so we are taking three or more sequences here and we are comparing them simply. So both we will learn in the next module. Till so far, thank you for watching. Please do like, share and subscribe the video. Also, if you have any doubt, you can ask it with you can directly connect it with me, or you can raise your doubt regarding the Google form.